Thank you. My name is Karen Miga. I co-hosted or led the discussion of breakout session 2 with Charles Lee. And I guess our question was really to, was to address how to start to study the hidden structural variations in the human genome. And so in summary, I'm going to share the slide here. There was a broad discussion and enthusiasm about how to use long-read sequencing to address these remaining gaps in the human genome, and that the study of the hidden structural variation could actually be useful in meeting that end goal of reaching a telomere-to-telomere phased assembly. There was a lot of discussion on how to analyze the hidden structural variation, and that new methods, approaches, and sequencing technologies just in general were necessary to meet that goal. Also, there was a lot of enthusiasm about completing more human samples. This was more leading to the drumbeat of creating a 1,000 genome type project. This is unlike the 1,000 genomes maybe that was put together with the short reads that focused on the single nucleotide variants. This needs to have a number of complete phased telomere-to-telomere genomes for structural variant characterization and getting into these parts of the genome that are otherwise difficult to characterize. Also, we need to move away from making simple computational predictions of these types of structural variants. New methods need to be developed, and so we can look at orthogonal validation of these new assemblies, and that the new standards should make it more cost-effective and efficient and available or able to scale to diploid genomes. And also, there was a lot of enthusiasm in our section about how to intersect the hidden structural variants with existing short-read functional data sets such as the ENCODE project. The main themes and priorities, we broke them into 3 groups. One was really emphasizing this need to improve comprehensive discovery, or to push on the technological developments needed for comprehensive SV detection. And this would bring into focus the satellite DNAs, the segmental duplications, the subtelomeric regions, the TE biology. All of this type of component should be kind of on the forefront of NHGRI's priority. Also, to do this, one might need to invest in new and improved methods to manipulate or deliver high molecular weight DNA for sequencing, with the ultimate goal of increasing throughput and sequence quality at the base level. Also, we need to support new sequencing technologies outside of the regular long-read players that we're used to thinking about, like Oxford Nanopore or even PacBio, giving an opportunity or a new space for new technology development to emerge that might actually be surpassing these existing technologies. And finally, all of this layered on top of this core concept of how we need to reboot the human reference genome. This is something that's been discussed in circles as the pan-genome project. This is the idea of moving into sequencing these high-quality reference genomes for not one or even a dozen, but moving into hundreds of individuals so we can begin to look at the resolution of the structural variation in the human population. This would be looking at high-quality, complete, diverse genomes for, structural variant detection and characterization. Now, how to move in this direction, there was some active discussion on that. For example, we started off saying, well, the haploid genomes, such as the heterodipteral mole or even the hap1, which is truly haploid, may be useful or helpful to begin to work into the space because you bypass some of the challenges that are offered by the paralogs or the two in the diploid genomes. But the clear goal, I think, that came out of the meeting or the breakout session was that we have to move towards these diploid genome assemblies, and that even though they're helpful and not fully utilized at the moment, we have to begin thinking of how we're going to actually utilize our skills, our technologies to start talking about diploids. And then finally, for function, there's an active discussion on how we can start to study the functional annotation in these particular regions. Maybe short read technologies or the technologies we've been employing for ENCODE are insufficient to really map into these dark hidden sequences of the genome. Maybe we need to have long read sequencing. Maybe we need to have new technological development to begin to study high-C maps, to begin to study these types of things that are often omitted because of mapability issues due to the short reads. Also, to bring in these types of short read data sets, also how do we begin to improve methods for imputation, and how can we begin to test this idea of what can be imputed and what can't? And that was something that we thought was a fundamental question that should be approached by NHGRI. So with that.